So now we've gone over in general terms how the neurotransmitter systems are organized and how to study them. So now we're just going to go through some of the major neurotransmitters systems and talk about how they're synthesized and packaged and the specific components of each one. So first we'll talk about acetylcholine. We've already mentioned that acetylcholine is the neurotransmitter of motor neurons, but also some neurons in the peripheral nervous system, including the autonomic nervous system, and some neurons in the brain too. And acetylcholine is synthesized from two precursors, this stuff called choline and something called acetyl-CoA. And those two are combined by an enzyme called choline acetyltransferase. This is choline acetyltransferase. It's abbreviated CHAT into acetylcholine. So the reaction looks like this. Don't worry too much about the exact structures. This is just to kind of explain where these things come from. So choline is this molecule that mostly comes from your diet. So you can synthesize it, but for the most part, it comes from the food you eat. So it's just found in your body and in your extracellular fluid. It actually gets taken up into the cell by a specific protein called the choline transporter. And in fact, for this reason, the availability of choline is what we call the rate limiting factor. So all of these neurotransmitters, and really actually all molecules that are made by your cells, have some rate limiting step or rate limiting part to their synthesizing pathway, because and it's that component that determines how much of it the cell can make. So for acetylcholine, it's the amount of choline, because again, that mostly comes from your diet and has to be taken up into the cell from extracellular fluid. Meanwhile, acetyl-CoA is a molecule that's just sort of part of a lot of different metabolic pathways, so it's always present in your cells at a certain amount. And so the enzyme, choline acetyltransferase, does what it says. So it transfers, that's what the word transferase means, an acetyl group to choline from acetyl-CoA. So the CoA part is sort of an adapter molecule that the enzyme breaks off, and then it just attaches the acetyl group. So this is the acetyl part, and this is the choline part, and that's where acetylcholine comes from. So again, this happens in the presynaptic terminal, and then the acetylcholine is packaged into vesicles by a specific acetylcholine transporter. This is a specific protein that, again, is only found in acetylcholine neurons. So you can use either the transporter protein or the enzyme as a marker for acetylcholine neurons. Again, if you were doing immunocytochemistry or in situ hybridization. But in either case, the neurotransmitter gets packaged like this, and then it gets released and it binds to its receptors, which are on the postsynaptic side. And we'll talk about receptors more later. And then in the synaptic cleft, there's an enzyme on the membrane of the presynaptic cell, usually called acetylcholine esterase, sometimes abbreviated ACE or ACE. And it breaks acetylcholine down back into choline and acetic acid. And this is the reaction down here. So acetylcholine over here getting broken down into acetylcholine, or into acetic acid and choline by a different enzyme now, acetylcholine esterase. Again, this is kind of unique to acetylcholine. Most other neurotransmitters get reuptaken rather than broken down. The choline can then be recycled, so it gets taken back up into the cell through the choline transporter. By the way, a lot of these transporters, many of them use ion gradients to kind of help move the molecule across. So the choline transporter and other transporters like it are what we call a co-transporter. So it moves a molecule of choline along with a sodium ion. So remember that sodium has a very large concentration gradient going down into the cell. So the choline transporter sort of makes use of that. It uses the movement of sodium to help move the choline also into the cell. And so that's how the acetylcholine gets broken down and recycled. Again, this all happens in the cell. And one of the reasons it's important to understand these steps is because if you want to study a neurotransmitter system, or if you think a particular neurotransmitter is somehow involved in, let's say, a disease, then knowing how it's made, how it's broken down, how it's packaged is important. So, for example, acetylcholine is involved, at least to some extent, at least in the early stages, with the disease Alzheimer's disease. So, Alzheimer's disease, you may know, is what we call a neurodegenerative disorder. So, it's caused by, or it's characterized by, the progressive death of neurons in the brain, and eventually it affects the entire brain. But the early stages, the earliest neurons to die in Alzheimer's are acetylcholine neurons. Now, it's not clear why, but they die first, and it does contribute to the early symptoms of dementia that are associated with that disease. And so, and at the time, at least that I know of, there's no real way to prevent that. There's no way to stop the neurons from dying, and there's no way to really bring them back once they've died. But one of the treatments for early stages of Alzheimer's is a drug, an inhibitor of this enzyme, acetylcholinesterase. So if you block this enzyme, then what you will do is prevent acetylcholine from breaking down. And if acetylcholine is not broken down, it will just stick around in the synapse longer. Eventually it gets broken down, but it's able to stick around in that synapse longer than it would otherwise, which basically just means that, again, even though acetylcholine neurons have died, those that survive can be more effective because each individual synapse is able to release more acetylcholine, presumably, or to keep it from being broken down as quickly. So again, it's not a cure. It just sort of is a treatment that may prolong the onset of some of the later symptoms of the disease. But again, it's important to know these steps so you can interact with these systems in this way. In fact, there's a whole class of drugs that are called ACE inhibitors or acetylcholinesterase inhibitors, and that's their job, is to help prevent the breakdown of acetylcholine. And then we have the catecholamines. So there are 3 catecholamines. Dopamine is one, norepinephrine, and epinephrine. And they're all called that because they have this structure here as part of their chemical structure called the catechol group. So if you know what this represents, this is a phenyl ring with 2 alcohol groups attached. And then if it's this is dopamine, that catechol group with just this ethylamine group attached, and then norepinephrine is has one OH, and then epinephrine has another methyl group attached to it. One note about the notation or the nomenclature for some of these things: norepinephrine sometimes goes by the name noradrenaline, and then epinephrine also goes by the name adrenaline. So epinephrine and adrenaline are exactly the same molecule. And in fact, their names mean the same thing. So the word epi means on top of or next to, and nephros comes from the Greek word for kidney, and that's because this hormone comes primarily from the gland that sits on top of the kidney called the adrenal gland. That's why it's also called adrenaline. But adrenaline, or the word adrenal, also means on top of the kidney. So one is Greek, epinephrine, epinephros is Greek, whereas adrenal, ad means next to, and renal means having to do with the kidneys. So that's the Latin name for the kidneys, or for the adrenal gland. They're, again, 2 different names for the exact same thing. One is preferred in kind of UK British English, that's adrenaline, and then epinephrine is preferred or more often used in American English, but they mean the same thing. Same with norepinephrine and noradrenaline. But they are, these all 3 together we call the catecholamines. And in addition to this structure, they also have a common synthetic pathway. So all 3 are derived from the amino acid tyrosine. This is tyrosine. This is one of the amino acids that your cells use to make protein. So all of your cells have tyrosine in them. But then neurons that make catecholamines will also have this enzyme called tyrosine hydroxylase. And tyrosine hydroxylase, again, every enzyme, most enzymes end in ACE, and usually you can kind of tell what the enzyme does from that name. So tyrosine hydroxylase adds a hydroxyl group to tyrosine, and this is a hydroxyl group, and that is the hydroxyl group that gets added. But this is not a neurotransmitter. This is, the long name is L-dihydroxyphenylalanine, because actually this molecule with no hydroxyl groups would be a different amino acid called phenylalanine, but usually it's just called DOPA. So L-dihydroxyphenylalanine is just called DOPA, sometimes just L-DOPA. So if you ever hear that term, that's all it means. And then a different enzyme called DOPA decarboxylase, again, does what it says it does. It removes a carboxyl group from DOPA. This is a carboxyl group. So by removing the carboxyl group, it turns L-DOPA into dopamine. And again, DOPA is still standing for deoxyphenylalanine, but now it's just called dopamine. And so if you're talking about a dopamine neuron, this is the end of the line. So a dopamine neuron will have tyrosine hydroxylase. Again, the rate limiting step here is tyrosine hydroxylase. So the enzyme tyrosine hydroxylase, its presence in the cell is rate limiting. So neurons that have tyrosine hydroxylase will make L-DOPA, and then if they also have DOPA decarboxylase, they will make that into dopamine. But the amount of dopamine you can make depends on the amount of tyrosine hydroxylase. And the reason this matters, again, is because if you want to manipulate this system, you have to usually go to the rate limiting step. So, for example, if you have a condition that's characterized by low dopamine, or you want to raise dopamine levels for some reason, adding more tyrosine won't help, because adding more tyrosine will not get you past the rate limiting step. You can either increase the activity of tyrosine hydroxylase, or what you can do is just skip that step. And in fact, giving L-DOPA to dopamine neurons will increase the amount of dopamine they make and the dopamine they release. And this is, in fact, a treatment for diseases that are characterized by low dopamine. The best example is Parkinson's. So Parkinson's is similar to Alzheimer's. It's characterized by a drop in or a death of dopamine neurons in the brain, particularly in a region called the substantia nigra. And again, you can't stop those cells from dying, you can't bring them back, but if you give L-DOPA as a treatment, it will get into the brain and it will increase the dopamine availability of the neurons that don't die. So again, it just sort of delays some of the longer term effects of the disease. But in any case, this is how dopamine gets made. And then it turns out that dopamine itself is actually the precursor for norepinephrine. So yet another enzyme. Every step here involves a different enzyme. This one's called dopamine beta hydroxylase. And again, it's going to do what it says it's going to do. It's going to add a hydroxyl group, this time to what's called the beta carbon of dopamine, which is this carbon right here. And by adding it, it makes it into norepinephrine. So that's the only difference between norepinephrine and dopamine, is that one hydroxyl group. And then one kind of weird trick here is that the dopamine neurons package dopamine into synaptic vesicles, which is exactly what you would expect. But then it turns out that norepinephrine neurons also package dopamine into vesicles, because this enzyme dopamine beta hydroxylase is actually inside the vesicles. So again, a dopamine neuron would not have DBH, but a norepinephrine neuron would, but it just happens to be in the vesicle. So what actually happens is norepinephrine gets synthesized in the vesicle instead of being packaged on its own. Now, if it's an epinephrine neuron, then it will have this enzyme, phentolamine N-methyltransferase, which again is a complicated sounding name for a pretty simple reaction. Basically, this methyl group gets added to the amino group of norepinephrine, and that's all epinephrine is. It's just a methylated form of norepinephrine. And now again, there's kind of some complicated shuffling here, because PNMT is found in the cytosol. So to make epinephrine, there's kind of a complicated pathway. So here are your vesicles. You have dopamine. It gets transported into the vesicle, and then it gets converted into norepinephrine. And then norepinephrine actually leaves a vesicle to eventually be converted into epinephrine, and then it goes back into the vesicle again, or into a different vesicle. And so you end up with this kind of complicated little dance that you need to make epinephrine, but that's just the pathway that it goes through. Again, all of these steps happen. So if you're in an epinephrine neuron, you'd be seeing all of these steps happen. Now you have, again, the reason to know about this is if you want to identify, let's say you want to identify a dopamine neuron, you can use tyrosine hydroxylase, because tyrosine hydroxylase will not be found in neurons that don't release dopamine, unless it's also it happens to be a norepinephrine neuron. So all three types, dopamine, norepinephrine, epinephrine neuron, all three will have tyrosine hydroxylase. So tyrosine hydroxylase is not always the best marker for that, for dopamine or any of the neurotransmitters in this category. But there are specific markers like the other enzymes or the specific vesicular transporters you can use to identify them instead. One more thing to note about the synthesis of this neurotransmitter, and this is again a property that's the case with a lot of neurotransmitters and a lot of biochemical pathways. The end product, epinephrine, and actually this is also true to the extent of norepinephrine and dopamine itself, all three of these actually go back and inhibit tyrosine hydroxylase. So that means once the cell produces a certain amount of any of these neurotransmitters, and the levels elevate enough in the cytosol, it actually inhibits the enzyme that produces it. This is called end product inhibition. And again, this is really common with a lot of biochemical pathways. Basically, it's a way for the enzyme to sort of regulate itself. So the product of the enzyme or one of the downstream products from the pathway go back and inhibit one of the earlier steps, just to make sure that the amount of that neurotransmitter doesn't get too high, or the precursor doesn't get used up too quickly. But again, this is another way that the levels of these molecules are regulated. Now, all the catecholamines, again, like most other neurotransmitters, are removed from the synapse by reuptake. So that means there's a specific transporter protein that removes these molecules from the synaptic cleft and puts them back into the presynaptic terminal. And again, this reuptake step is an important thing to understand, because this is an important place where you can manipulate these neurotransmitter systems. And in fact, there are a whole bunch of drugs that act as catecholamine reuptake inhibitors. Cocaine and amphetamine are probably two of the most well-known. So again, they work by blocking the transporters that would remove those molecules from the synaptic cleft, which means they stay around in the synaptic cleft longer, which means that those synapses are just more effective. And then in the case, especially dopamine, is involved in your brain's reward system. So a lot of drugs of abuse, like cocaine and amphetamine, work either directly or indirectly by raising the amount of dopamine either released, or again, in this case, preventing it from being taken back up or removed from the synapse. Okay, now serotonin is a totally separate type of neurotransmitter. It also goes by the name 5-hydroxytryptamine. So in fact, a lot of the times you'll see it just abbreviated 5-HT. That's what that stands for. In fact, the receptors for this molecule are called 5-HT receptors, sometimes instead of serotonin receptors. Serotonin is made from, again, like the catecholamines, an amino acid, but this time an amino acid called tryptophan. Again, tryptophan is found in your cells because it is one of the amino acids that your cells use to make proteins. And tryptophan in a serotonin neuron is broken down by an enzyme called tryptophan hydroxylase. And tryptophan hydroxylase, again, you can figure out from the name what it does. It adds a hydroxyl group to the tryptophan. And so this molecule here is called 5-hydroxy tryptophan, 5-HTP is how it's abbreviated. And then another enzyme called 5-HTP decarboxylase removes the carboxyl group. This is a carboxyl group from 5-HTP, and you end up with 5-hydroxytryptamine, which again is just the long name for serotonin. So serotonin has just a two-step pathway, two enzymes. The tryptophan hydroxylase is actually rate limiting, but you can actually manipulate it by by the levels of tryptophan. In fact, serotonin, we'll learn later, is involved in, among other things, your sleep-wake cycle. So the parts of your brain that sort of keep you awake and put you to sleep at night have serotonin neurons as part of that network. And you can manipulate serotonin levels, it turns out, by raising tryptophan levels. And tryptophan, being an amino acid, which is found in protein, can be elevated in your system by eating lots of protein. So if you have a high protein diet, you might have a high tryptophan level, and that's supposedly, although this may not be actually true, but the old saying goes that if you eat lots of meat at a meal, like let's say turkey at Thanksgiving, then you raise your tryptophan levels, and that by raising serotonin can give you that effect of feeling really sleepy after a big meal. Again, the actual science behind that is not exactly rock solid, but it does kind of illustrate the point that this is how one of the ways in which these neurotransmitters and their precursors and their biochemical pathways can be influenced. And then serotonin, like catecholamines, are removed by reuptake. Again, this is an important step because you can effectively raise the activity of these neurons by blocking those reuptake inhibitors. Another thing that serotonin is involved in is various mood disorders. So things like depression, bipolar disorder, for example, in some people are associated with lowered levels of serotonin or lowered serotonin activity in neurons. And so one of the best known treatments for those mood disorders are the serotonin reuptake inhibitors, in fact, they're often just abbreviated, SSRI, which stands for selective serotonin reuptake inhibitor. So some really well-known pharmaceuticals like Prozac and Zoloft, those are all drugs that specifically block the serotonin reuptake transporter. So again, that's why it's important to understand these pathways. One more enzyme that's worth mentioning. So again, all of these are removed from the synapse by transporters, but once they're in the synapse, two things can happen. Either they can get repackaged into vesicles, or they can get broken down. And actually, all of these are broken down by the same enzyme called monoamine oxidase. And again, just like with acetylcholine, even though this happens inside the cell instead of outside, this is another way you can manipulate these neurotransmitters. So there's yet another category of pharmaceuticals called monoamine oxidase inhibitors, or MAOIs. And those are drugs that specifically block this enzyme and, again, prevent it from doing its job, which means that all of these neurotransmitters are in their neurons are elevated. And so again, MAOIs are also sometimes used to treat conditions like depression and other what we call mood disorders. Now, the amino acid neurotransmitters, glutamate, glycine, and GABA, are a little bit simpler because especially glutamate and glycine, you don't need a special biosynthetic pathway for those. There is one, so they're both synthesized from glucose. Cells can make it from glucose, or you can just get glutamate and glycine from your diet. Those are both amino acids that are found in every cell in your body, and in fact are found in every cell of every organism. So whenever you eat anything with amino acids in it, you're going to consume some glutamate and glycine. So you don't need, there is no specific enzyme that makes glutamate or glycine in neurons. So you can't use the synthetic enzyme as a marker for these neurotransmitters. But you can look for their specific vesicular transporters, because there's a specific glutamate transporter and a specific glycine transporter, and those are only going to be found in glutamate neurons and glycine neurons respectively. So if you want to know if a neuron releases glutamate, instead of looking for glutamate enzymes, you would look for the glutamate transporter. GABA, meanwhile, is also an amino acid, even though it's missing one of its carboxyl groups. But really, it's just a decarboxylated version of glutamate. And so GABA is made by an enzyme called glutamic acid decarboxylase. Again, it does what it says on the box. So glutamic acid is just another name for glutamate. Glutamate if you know your chemistry, is just the conjugate acid or conjugate base, I'm sorry, of glutamic acid. And so GAD, or glutamic acid decarboxylase, just removes this carboxyl group, and you're left with gamma aminobutyric acid, which is what GABA stands for. Again, you can use GAD as a marker for GABA, or you can use GABA transporters. Now, these neurotransmitters also are much more common in the nervous system. So glutamate and GABA especially are the main excitatory and inhibitory neurotransmitters, respectively. So glutamate's mostly excitatory, and GABA's mostly inhibitory. And so most of the circuits in your brain have some glutamate or GABA neurons in them. And so there really aren't as many pathways, molecular pathways involving these neurotransmitters that are directly related to any major diseases, because if you have a problem making glutamate in your brain or releasing glutamate in your brain, probably you're not going to survive very long anyway. So these aren't really connected to those more subtle conditions like depression or anxiety or so on. Now, before so those are the major kind of classical neurotransmitters. And again, we talked about what these strict criteria are for being defined as a neurotransmitter. But there are other molecules that neurons make and release that in some ways act like neurotransmitters, or at least they allow neurons to communicate with each other, even though maybe they don't fit all the strict definitions of a neurotransmitter. So an example are these molecules called endocannabinoids. So these are molecules that are produced by certain neurons, and they're these big, long hydrocarbon chains. In fact, they basically act kind of like lipids. They have a long non-polar region, which means they easily cross the membrane. And in fact, they don't get packaged into vesicles. Instead, neurons that produce them, the molecule just crosses the membrane, and then it just diffuses over to the neuron that it's going to have its effect on. And then there are receptors for this molecule called cannabinoid receptors, or in this case, here it's called CB1, but that just stands for cannabinoid. And these receptors then sort of they're G protein coupled receptors, and they trigger an effect in the cell. So there are a couple of things about endocannabinoids. First of all, they're synthesized in neurons that are experiencing high activity. So this neuron down here will make neurotransmitters in response. The enzyme that makes endocannabinoids is triggered by calcium. So calcium activity triggers the production of endocannabinoids. Calcium activity is usually the result of a lot of depolarization. And so in presynaptic or postsynaptic neurons especially, there are a lot of calcium channels that may be voltage gated. And so this is one way for that calcium to have an effect. It triggers this enzyme. And then like I said, the endocannabinoid itself diffuses across. It doesn't get packaged into vesicles. It activates a specific receptor though. And through its G protein, it actually inactivates calcium channels on in this case, the neuron that receives the cannabinoid signal. And so in a sense, these are actually what we call retrograde neurotransmitters. So retrograde just means backwards, which means that instead of this neuron up here, we consider the presynaptic cell, because it does have vesicles and that contain some neurotransmitter, usually an excitatory neurotransmitter. And then this cell we call postsynaptic because it has receptors for that neurotransmitter. But the endocannabinoids go the other way. They go back to the presynaptic terminal. And so the function probably of these endocannabinoids is to act like a feedback signal. So essentially the endocannabinoids tell the presynaptic neuron, especially if it's been highly activated, to kind of quiet down. And this again is common in a lot of circuits. You have these sort of feedback loops between two sets of neurons. Another interesting fact about endocannabinoids, the reason they're called endocannabinoids, the prefix endo means endogenous or internal, and cannabinoid comes from the word for the marijuana plant, cannabis. I'm spelling that right. So cannabis, of course, is the plant that produces a molecule called tetrahydrocannabinol, which is better known as THC. And so THC, of course, is the active ingredient in marijuana, and THC also activates the cannabinoid receptor. That's why it's called the cannabinoid receptor. And this is actually an example of a receptor that was discovered before its ligand was discovered. So THC, of course, is not found in your brain normally, and it's only found if you put it there, but it does activate the cannabinoid receptor. So, of course, it wouldn't make a lot of sense for your brain to express a receptor for a molecule that isn't normally found in your brain. So anytime this happens, anytime a drug is identified and then a receptor is found in the brain that binds to it, then we start looking for the ligand, the endogenous ligand that it binds to. In fact, at this point, when you have a receptor that binds to a drug like this, an exogenous drug, we call, so in this case CB1, we call it an orphan receptor, because we know that it's a receptor, we know that it binds to something in the brain, but we don't know what. We don't know what the ligand is. So until it has a ligand, we call it an orphan receptor. And you can also have orphan ligands, so molecules that were released and have some sort of effect on a postsynaptic cell or some other cell, but we don't know what the receptor is. It just so happens that this one binds to THC, and so the receptor got called the cannabinoid receptor because THC is also a cannabinoid, but it's an exogenous cannabinoid. And so then once a molecule is found in the brain that binds to it, that got called endocannabinoid. So that's just where some of these names come from. And of course, it also kind of explains the effects of THC. So the cannabinoid, endocannabinoids are generally inhibitory signals. They reduce the activity of the brain, and so that kind of explains how THC does its job too. It sort of reduces activity in the brain overall. So endocannabinoids are one type of sort of non-classical neurotransmitter. Another class are the purines. So if you know anything about biochemistry, you know that the purines are a type of nucleic acid. So there are actually two sets of purines. Adenosine is one, the other is guanine, but adenosine in particular and its phosphorylated counterpart ATP both can act as neurotransmitters. ATP, it turns out, is generally an excitatory neurotransmitter, and then it's often a co-transmitter too. It's often released with other neurotransmitters at the same synapse. Adenosine, meanwhile, actually is inhibitory. So it binds to a different set of receptors, and those receptors are inhibitory. One interesting thing to note is that the adenosine receptor is antagonized by caffeine. So caffeine is, of course, found in things like coffee and soda, and it works by blocking the adenosine receptor. Now, this kind of explains the effects of caffeine because, again, adenosine receptors are inhibitory, so caffeine is often thought to increase brain activity and increase sort of feelings of alertness and wakefulness, and so it probably works this way by blocking those receptors. In fact, probably the job, or one of the jobs of the purine receptors, is to let the brain have a way to measure the general energy levels that are found in the tissue, because you probably know that cells that convert things like sugars into energy, or convert the energy into ATP, and then that ATP, the energy that is stored in that molecule, is used by most of the enzymes in your cell. And so if you're low on energy, if you're low on ATP, or if you have sufficient ATP, then it makes sense that the ATP receptors would be excitatory, because that would be a signal to the brain that there's plenty of energy available, and so it should maintain high activity levels. Meanwhile, adenosine is one of the byproducts of ATP breakdown. So as ATP is used, adenosine levels go up. And so that's the two are part of sort of a cycle. So as one goes down, the other goes up. So high levels of ATP, again, means plenty of energy available. High levels of adenosine, by implication, means low levels of energy. And so the presumption is that adenosine receptors in your brain basically tell your brain if you are low on energy. And so high levels of adenosine will cause sort of sleepiness and sort of triggers your brain to enter shutdown mode so that you can rest and restore your ATP stores. Caffeine, meanwhile, presumably has its effects then by blocking those receptors. So in a sense, caffeine is sort of tricking your brain into thinking you have more energy than you really do, which is why caffeine temporarily can make you feel more awake and alert. But after long periods of time or elevated caffeine levels, you can have a big crash from it. You can be much more tired than you would have been if you had not taken it. And then there's a bunch of molecules that, again, aren't really clear if they're really neurotransmitters or not, but have some properties or have some ability to let cells communicate over distance, and so they can sort of be classified on the boundaries of what we would consider neurotransmitters. So, for example, there are several small gas molecules like nitric oxide and to some extent even carbon dioxide and carbon monoxide that are maybe not strictly neurotransmitters, but do allow some communication between cells. Carbon dioxide is probably the most straightforward one because all of your cells produce carbon dioxide as a byproduct. So as you break down carbohydrates in the presence of oxygen, your cells produce CO2. Most of that CO2 gets exhaled when you breathe out. But there are also proteins in some cells, including in your brain, that can measure the CO2 levels, or either directly or indirectly. And they then can, so there's a place in your brain called the hypothalamus that measures CO2 levels, and it can then monitor sort of how well oxygenated your blood is, because if your CO2 levels get too high, that means you need to increase your respiration, for example, to get rid of CO2. So there are, in fact, neurons that respond to it. Carbon monoxide also can be found in your body. It's not clear if it actually really is a neurotransmitter, but nitric oxide definitely is. So nitric oxide, for example, one of its roles in the body is as a vasodilator, which means that it can trigger blood vessels to dilate and let more blood through. And that can be also controlled by the brain. Just an interesting side note about nitric oxide: one of the targets of nitric oxide, one of the downstream molecular targets within its pathway, is broken down by an enzyme, which in turn is blocked by the drug Viagra. So Viagra actually works by interacting with the nitric oxide pathway. Again, just another example of why it's important to know about neurotransmitters and study the pathways of these systems. Probably one of the most financially lucrative drugs ever invented, Viagra. All right, so we will stop there for now, and we will come back next time and talk about neurotransmitter receptors.